let's go!
How to make
There we
it
go.
be exactly the right way and the science behind that. Or it could be how to build your house in a breathable way that would be more naturally appropriate. Or it could be something really important that we don't talk about enough in this culture, the microbiome and how we relate to that, which is a whole...
Enviromes.
We did a whole compost thing last year. As near as I can tell, this is not that different. So we're going to... It's the same but different, man. So
very good.
To Alan Booker,
Thank you.
He can decide which of his letters he explains to you if we need letters.
Very good. We're making certain we've got signal. Oh, I got signal now? I'm seeing signal.
Yes,
good.
Yes.
All right. We got microphone signalage. So this morning we're going to be doing we're going to break the session in half. We're going to do the first hour on microbiomes, viromes, and human health. And the second, we're going to take a 15-minute break. We're going to come back, and then I'm going to talk about EMF exposure. I started out, my undergraduate degree was in electrical engineering, so I can get pretty geeky there if we want to. But with this group, I'm always trying to figure out what level do I approach this at, because usually at the Institute, what I'm doing is we're teaching engineers and architects and people with, who might be coming in as grad students and so forth. And so a lot of time, what we're doing is we're teaching, I might be teaching this to medical healthcare practitioners and stuff like that. So let me sort of pop over here so I can get my remote control going. There we go. So I do have this for you. Tomorrow I'm going to be covering... going for an hour on soils, and then we're going to have lunch, and then we're going to come back and we're going to do soil microscopy for an hour. So I'm like, sure, Nicole, I usually spend 3 on soils, maybe 4, and then soil microscopy can be another 2 days, but we'll do it in 2 hours.
We'd come here for a week later this year. We'd probably get some students.
Yeah.
Yeah.
So, what I want to do is this for this group. I did something on soils last year, and we talked about soils, and we talked about how plants uptake nutrients from the soil, and we went through all of that. So what I want to know from everybody, I have hundreds of slides on soil. I can talk about all kinds of crazy stuff. So what I've done is, since I've only got an hour, to the rest of today, I want you to be thinking about what do you want me to talk about on soils tomorrow. And then I will just pull the slides and talk about those things, or I can do a quick review of what we talked about last year and just go deeper into certain aspects. Then we will pull out the microscopes and we will have some fun doing some soil microscopy and see if we can actually look at the little critters in there, coming out of the soil. But today we're going to start with microbiomes, viromes, and the human health right now, which will be cool because when Sandra is here right after lunch, we'll be working with them live. Okay.
Like that was intentional.
Yeah. So before we jump in, how many of you? Let's just kind of see where we are. How many of you could give me a good, solid definition of a microbiome? Okay. How about a virome? Oh, cool. Okay. So most people haven't heard of viromes. We'll get into that. And if we talked about the human microbiome, which one do you probably think of the most? The gut microbiome. But there's all these others.
I know.
Right? So we're going to talk about that as well, because it turns out they're all important. And so what I'm going to do for you today is you're going to see a couple of tables, technical tables that I went ahead and put in for this presentation. These technical tables that I'm pulling out are tables that I created for the biocompatible design standard that's going to be being published in a couple months. And so we're going to dig into the deep side of it at the end when I pull out the technical tables. But it will give you the technical information for designing environments that are deeply compatible with life systems. Okay? So with that, we're just going to jump in and talk about microbiomes, viromes, and human health and how it all works. And one of the fun things is I'm currently working with a medical doctor who she lives on an off-grid farm, has for years, And she is very interested in teaching medical practitioners about how soil microbiology and the soil microbiome impact human health and help people understand how nutrient-dense food, but also the microbiome of the soil that you're around impacts human health deeply over long periods of time, right? All right. So how many of you had high school biology or college biology? Yeah.
It changed between high school and college.
Exactly. It changed and it changed again. And the reason for that was, and we were talking about this last night a little bit with some folks, was that early on, the way we categorized life was what phenotype, which basically means you look at it and you see what does it look like, what's its shape, what's its function, right? And then the Linnaean binomial system of, saying that, wormwood is Artemisia absinthium is its scientific name. That whole system started out by looking at plants and animals and microbes and saying, well, this one looks like that one, and it seems to function kind of the same way. So we're going to actually create the whole tree of life based upon that phenotype. But then large-scale genetic analysis became available, and we were able to start looking at the genome, the actual genes of all these different species, and we started realizing that was very misleading in a lot of cases. What you could see is, well, this thing looks like that, but if you look at its genetics, this is way more related to something way over here than it is to the thing that it kind of looks like. And so there was this big transformation in biology where there was an argument for a while, like which way are we going to categorize everything in the tree of life? And what it turned out was that the genotype folks are winning. Because I'm not saying they won, because it's still changing. Like, you have to keep up with it because there's a whole debate, right? So they came up with this idea. It used to be, when I was doing college biology, we had kingdoms were the top, right? It was kingdom, phylum, class, order, family, genus, species, right?
That's what I'm for.
Right? Well, now we put domain up top, okay? And what they've come up with is they realized that what we used to call bacteria, put it all in one big thing, it isn't just one big thing anymore. They actually changed it, and it's now two different domains, bacteria and archaea. And then we have all of eukarya. And the eukarya are, of course, let's go back and remember, what's a prokaryote? No nucleus, that's right. It's a single-celled organism that has no bounded nucleus. What is a eukaryote? It has a bounded nucleus. The primary genetic material of that cell is inside of a nucleus. Now, why did I say primary? How many sets of DNA do you have? Sets. Distinct sets. What was it? Two. You have two sets. Okay. What are the two sets of DNA you have?
Nucleic and mitochondrial.
You have your nuclear DNA. That's the stuff that got made by mom and dad, their DNA coming together, right? And that's in the nucleus. But then you have the mitochondria, which is a little organelle. That's the powerhouse of your cell, right? And that has DNA. Where does that DNA come from? Mom. That comes just from mom. You get her mitochondrial DNA, which means that you can actually do DNA analysis two different ways on a human being. Here's a good one. How many sets of DNA does a plant cell have? 3. Has nucleus. It's a eukaryote. Okay. It also has mitochondria, but it also has chloroplasts. And the chloroplasts also have their thing. And I know this is way off in the weeds, but what happened was, back in the 20th century, Lynn Margulis, an evolutionary biologist, proposed something quite interesting. She said, and she called it symbiogenesis. She said what happened was, back in the old days, you had these simpler prokaryotes, and you had some of these eukaryotes, more complicated cells. And what happened was that every once in a while they would realize, the prokaryote would realize, life out here on my own is kind of tough. Why don't I move in with a roommate and actually just move in? So they would go inside of a larger eukaryotic cell. And so her proposal was that our mitochondria originally started as free-floating prokaryotes. And then later, they became part of our cell. And the same thing happened with plants, with mitochondria and with chloroplasts, right? So what's happening was that we got these three domains now. Bacteria and archaea, they got split in half. They used to all be bacteria. Archaea, why do you think they called it archaea? It was old. This is some of the oldest forms of life. And then the bacteria, and they're very distinct. And then in eukarya, there's animals, there's plants, but then there's also ciliates, flagellates, amoebae. Those are our protists, and those are part of the microbiome. And then fungi, which are also part of the microbiome, right? So when we talk about a microbiome, these are micro, of course, means we have to look at it through a microscope to see it. And biome means a community of life. So a microbiome would be a coherent community of life that forms its own ecology in a certain environmental niche. And that's what we mean by microbiome, right? So definition of a microbiome, the simplified definition is a microbiome is the collection, excuse me, of all of the microorganisms of various species, including bacteria, archaea, fungi, protozoa, etc., that colonize a specific environmental niche. Okay? And it turns out that this is so fundamental to life processes that the health implications for humans are vast. Okay? And we have what are called nested microbiomes. Where we're sitting right now, we have the environmental microbiome, which is basically, Nicole is setting up really well. She's got plants and animals and trying to generate really healthy soil. And if you do that well, it creates a very healthy environmental microbiome. Then we have what we would call the building microbiome, which is the microbiome that is on the surfaces and in the walls and in the cracks and crannies of this building. And that's nested inside of the environmental microbiome. And that can either be healthy or unhealthy. And then at the moment, all of our microbiomes on our bodies are nested inside the building microbiome, which is nested inside of the environmental microbiome. And that has a profound impact on our health, moment to moment. Because we are beginning to suspect, have pretty good evidence of this, that our own microbiomes collectively probably account for 70 to 80% of our immune function. So if we have dysbiosis, which means bad health of our own microbiomes, then our immune system is big trouble. If we have really healthy microbiomes in our body, then our immune function is typically much better, right? And we talk about the gut microbiome because a lot of research has been done on it. It was one of the ones that it helps us digest our food, right? It intermediates between us and the outside world with what we're drinking and what we're eating and so forth. And so a lot of research has been done in it. And we'll talk about it a little bit more as we go, okay? But how about a virome? Okay, this is where it gets interesting. A virome is the collection of all of the viruses occupying a single organism, holobiont, or ecosystem. We'll come back to holobiont in a moment, okay? So back in the 1970s, 1980s, some of us are old enough to remember when bacteria are bad and you need to kill them all, right? Anybody remember that? Yeah, they're all
So much more so.
right. And part of the reason for that, of course, was the only time we were paying attention to the bacteria was when they were causing a problem. And all the research dollars went into like, oh, yep, that bacteria is a problem under this circumstance. How do we kill it? Right? Oh, that bacteria is giving us problems. Kill it. And so pretty soon the idea was like bacteria bad, kill them all, right? And then the research started to emerge that said, oh, no, wait. The bacteria are doing, and eventually archaea, when they divided them up into two different categories, those are doing some profoundly important ecological functions, including our own health, including our digestive system, right? It's like, as a matter of fact, let's just put it to you this way. What would happen to a human being if we removed their microbiomes? They would be dead. That's right. Okay? You literally cannot live without your microbiomes, right? Okay. So a virome, however, we're still at the same point with viruses that we were with bacteria back in the 1970s and 1980s, right? Oh, viruses are bad, right? Destroy them all. And notice I didn't say kill them all. Why not? They're not, at least, okay, there's been some debate. Are they alive? Are they not alive? And so, given the definitions of life that I use, they are not alive, okay? Because what they are is they're little packets of genetic information that can go into living cells, and that information can be used. Now, at this moment, every one of us has several trillion viruses in our body. That's our baseline state, right? Now, at the current, a lot of scientists would tell you, oh, yeah, those viruses, they're just all bad things. It's like, well, then why does nature keep on producing them in the trillions and trillions and trillions? And the research is now just emerging, just like I was at some of the forefront of the microbial and microbiome research. I'm now pushing on the health implications of the virome. Because, and this will really blow your mind a little bit, what do we think the virome actually does? Well, the evidence starts to point to the fact that these are genetic messengers that go back and forth between living cells. So, we can get something called horizontal gene transfer. Who knows about horizontal gene transfer? Okay. Very good. Horizontal gene transfer is this. Let's say that a bacteria is subjected to something really, really awful, glyphosate. Okay? Glyphosate blocks a metabolic pathway that, when it's blocked, kills all the plants, but also tends to kill the bacteria and the archaea, because it's like it blocks, if it's life, it blocks that metabolic pathway, and therefore it'll kill the bacteria just like it'll kill the plant cell, right? So the bacteria is like, well, this isn't good. We need to figure out a way around this. And so finally, one of the bacteria did. Says, oh, I figured out an alternative metabolic pathway that I can use when I've got glyphosate around. And so it did that. And then it says, like, well, I'm just going to teach all my buddies how to do this too. That's called horizontal gene transfer. Here's the genetic information that I figured out how to adapt to the emerging environmental situation. And here it is, because bacteria are cooperative. That's a whole thing we can get into with soil and multi-species quorum sensing and all that kind of stuff. But it turns out that in order for them to thrive, they need their the rest of their buddies to thrive too, right? So they started figuring out how to tell everybody else how to survive glyphosate. Well, along comes Monsanto, back when they were still Monsanto and not bought out by Bayer, right? And finds in a ditch where there's runoff from an agricultural field where they're spraying it with glyphosate, they find bacteria that can survive glyphosate. And they find the genes and they splice it into plants and they create Roundup Ready soy and wheat and so forth. And now the wheat and the soy can survive the glyphosate, right? But, nature, not to be outdone, does what?
It changes again.
It does horizontal gene transfer between those bacteria and the plants, and we get super weeds. So not too long after that, we start finding weeds that are glyphosate resistant, and now there's scientific evidence that shows that horizontal gene transfer happens between bacteria and plants. right? The bacteria taught the weeds how to survive glyphosate. Okay? Now, we're starting to suspect that it's actually viruses that are moving back and forth that are these little packets of genetic information that are being used to intermediate parts of this horizontal gene transfer and allow life to adapt faster than it could. Because for a long time it was like, hey, Jack, it's all just random mutations and life adapts just by random mutations. And you're like, is that fast enough to adapt? No. It takes generations, right? So what if life had come up with this like sharing and cooperating strategy? Hey guys, I figured out how to solve the problem. If I share it with all of you, our whole community can thrive. What do you think? And that's what it seems to be. So where we are is we're starting to understand that viromes shouldn't just be destroyed. That viromes should be, just like we're going to talk about today, the best way to have it is to avoid pathogenic bacteria and archaea and fungi is to have all the good guys present and really healthy. Then the same thing is true to having a very positive virome. Okay? So we can start getting out of the idea of just like sterilize everything, destroy all of the bacteria, destroy all of the archaea, destroy all the fungi, destroy all the viruses, and so forth. Yes, it is. It really is. Yeah. Okay. So let's talk about the human microbiomes then. I should have made that like plural. Okay. Human beings do not host a single microbiome, but live in relationship with a constellation of distinct microbial ecologies distributed across multiple body domains and life stages. These ecologies differ in composition, function, and environmental sensitivity, yet all help shape immune function, metabolic regulation, barrier integrity. What does that mean? Barrier integrity.
Protection.
Your skin barrier, your gut barrier, right? Like your mucosal membranes, so that like when your food comes into your gut, what gets across the membrane into your bloodstream and what doesn't, that's barrier integrity. Developmental patterning and the body's ongoing exchange with the larger living world, okay? So one of the things to understand is that we are in constant communication with our microbiomes. One of the ones that's most studied is the gut microbiome and what Dr. Natasha Campbell-McBride ended up calling the gut-brain axis. Because it turns out that the microbes in your gut can send signals up the vagus nerve to the brain, right? This is why if you get gut dysbiosis, because you have bad bacteria in your gut that want sugars, and you stop eating sugars, what do? They go grab the vagus nerve and they go like this, hey dummy, send more sugar. Why do you think you get sugar cravings? It actually comes more from your gut microbiome than it does from your own brain, And it turns out that you have more, and you'll hear this said, like you have more cells by number of microbiome than you have human cells in your body. Okay. Now, these microbial cells are tiny compared to human cells, which means that your whole microbiome virome probably accounts for somewhere like 4 to 5 pounds of your mass right now. Okay. But in terms of sheer number of cells, they outnumber our cells. Okay. So when you have
We're less us than us.
yes, right, by a sheer count of cells, right? Okay. So so let's talk about a holobionte for a moment, right? Because when you are doing any sort of ecological design or health design or design that understands life, you have to understand what a holobionte is because you are a holobionte. Yes, you are. Sorry. Sorry. Okay. A holobionte is a superorganism formed by a large host organism, in our case, it's Homo sapien, right? living along with all the other living organisms living symbiotically in, with, or around it. A human holobiont is formed by a single person along with all of the associated microbiome and virome. Okay? And this happens to be a diagram that I got of a poplar tree and all of its associated different forms of microbiomes. And the whole thing is this poplar tree cannot thrive without all of those symbiotes. Therefore, it is technically better to think of the poplar tree as a holobiont. Instead of thinking of it as that's a poplar tree, you think of it as that is a consortium of living beings, with the poplar tree being the backbone and all these other things going along with it, and that all together can thrive. And you're the exact same. That's what I was saying. If you don't have all your microbiomes, you literally will die. Therefore, we are a holobiont, which is why I can definitively say, yes, you are a holobiont, because without your symbiotes, you literally can't live. You would be dead in short order. Okay? So this is a very useful concept. We were doing a whole thing, we're going to plant a billion trees. And I was like, if you do that, you're going to fail. We need to actually plant.
Your trees will die.
The trees will die. They'll actually die. A large percentage of them will die. I'm like, what we found is if you want to plant trees, you want to plant tree holobionts. In other words, you want to put the tree in along with making certain it has all of its partners that it needs to thrive. Otherwise, it's like the tree is struggling from the very beginning. And you'll have a huge amount of fallout. You'll lose like 70% of the little bare roots you put in certain environments, right? Anybody ever tried that before? Yes.
One of the reasons why if you plant a tree in your front yard, it probably doesn't do very well.
Right.
It doesn't have its microbiome there.
That's correct. Yeah, and so trees typically have a lot of beneficial symbiotic relationships with fungi. And, if you have a monocrop grass lawn, we could look at that under the microscope and it looks like the surface of the moon. It has bacteria, some bacteria, but almost nothing else. So, yeah. So when we did this at a university, I took over the cell biology lab and we were doing this with all these like PhDs in biology and their grad students. They had never seen anything like it before because they were used to looking at, under the microscope, cultures on a sterile agar substrate where they would know what they were culturing. And all of a sudden, I'm giving them samples that have literally thousands upon thousands of species of bacteria and fungi and everything else. We go get some soil from right underneath the grass in front of the building, and we look at it, and they're like, "Where's all the life?" I'm like, "Exactly." Yeah, this is what happens when you grow grass with chemicals, right? It kills everything else.
Are you
Yes.
dipping for an inoculant? Are you spraying? What is the inoculant? Do you have to do
I
by species?
prefer to actually plant seed than to, because the plants will almost always be more robust if they germinate in place. And so what we like to do is actually pre-inoculate the seed. And there are now little machines that will create micro seed balls that are optimized for the particular species that you want. And then you can deploy them with drones. And this is actually really helpful for reseeding forests because you can get over terrain, right? And then you can allow for the seeds that are in optimal locations to germinate and so forth. And you want to do that with stages because you don't want a mono-aged forest, right? So you start off, you seed, you let some things get up a little bit, you come back, you seed again, because we want to actually seed a multi-aged forest, not because mono-aged forests are not healthy. Yeah.
Seth old school will sell trees, but he won't sell them without the dirt on them.
Yep.
Yes.
Right. And he won't sell them over 2 years of age.
Yes.
From a practical standpoint, if we're doing this small scale on our own property,
Yeah.
to acclimate the tree to the biome, is a small amount of compost in the hole from that, specifically from that property,
Yes.
and maybe compost tea sprayed on the leaves of.
Yeah. You want to foliarly spray compost tea on the leaves because it's just as important to get the good microbiology on the leaf surfaces as it is to get it on the roots. This is especially here in the southeastern United States or in high humidity environments, where if you don't have that, you can get fungal diseases on leaves. Competitive exclusion, thank you for transitioning into my next slide, is like the way to handle this. So what in the world is competitive exclusion, right? In both buildings and bodies, stable microbial communities help occupy ecological niches before opportunists can take hold. Our skin, on the gut, beneficial microbes suppress pathogens by competing for space, nutrients, and chemical advantage. In buildings, diverse and well-regulated microbial communities may help resist domination by opportunistic problematic organisms, especially when moisture is well controlled in the building. When ecosystems are disrupted, whether by antibiotics, oversanitation, or chronic dampness, open niches appear and unwanted microbes can expand. Okay, so this has to do with right here. Let's see if we can get it. Yeah, barely, I can't really see that. Okay, Gauss used cultures of protozoans to develop competitive exclusion principle in 1934, and he stated that two species cannot coexist on the same limiting resource. Right? In the same ecological niche, one is going to outcompete the other. And so our idea of how to keep pathogens out, instead of try to sterilize everything, which never works, because as soon as you sterilize it, just comes right back, is instead get a good guy in there, right, that will take up that resource, okay? I sometimes use this analogy. Imagine we had a big condominium complex in a big city, right? And we're like, you know, hey, Nicole, we don't want the drug dealers to take over our, you know, what do you do? Well, I tell you what, we'll put up machine gun emplacements outside of the condominium, right? And we'll shoot anything that comes close to it. What do you think?
The mailman's going to be really sad.
Exactly. Exactly. Do you think it's going to work? No, they're going to find like how to sneak past the machine gun. Next thing you know, you're going to have you're going to have machine gun nests, you know, and drug dealers inside the building, right? This is the sanitation hypothesis. Kill it all. Never. If we went and found a lot of really good conscientious tenants and filled up the entire building with them, and they took care of the building, they cleaned it, they took pride in it, and they said, this is our home, we're going to defend it. What do you think?
That's a
better solution, right? And what if those particular tenants happened to know about the bad guys and had very specific, could tell the difference, and very specifically had tools to make certain the bad guys couldn't get a foothold? That's exactly how it happens microbially. That's what competitive exclusion is. If we fill up an environmental niche with good guys, they all do their own literal chemical warfare. They create very precisely targeted defenses against pathogenics that they don't want there. And so competitive exclusion is the way we tend to want to have as our primary way of maintaining environmental health. Yes?
Sounds a lot like how fermenting works, where the
Yes.
good guys outcompete the bad guys.
Fermentation, like what Sanders going to be showing us, is exactly that. It's competitive exclusion, right? We put a little salt in there to slow down the bad guys a little bit, and then the lactobacters and acetobacters and lactoferments get going. They start to create conditions that the bad guys can't tolerate very well, which is acidic in this case, and they competitively exclude the bad guys while also making the nutrients in the food more bioavailable to us, and therefore preserve the food while also making it more nutrient available for us, right? So yes, that is a form of competitive exclusion. Here's what you see, right? Here are two different groups that were grown separately, and you can see their populations come up. But when you grow them in a mixed culture, one succeeds and the other doesn't. And that's competitive exclusion, okay? All right, so let's get to Principle 6 in the biocompatible design standard, right? The formal title of this is Recommended Practices for Biocompatible Design. Okay, and I'm and Oakland gets to hear way too much about this because I'm in the final processes of pushing through to get it published. But principle 6 is this: nurture healthy microbiomes and viromes in all ecosystems. Okay, that's the principle. Now, inside of principles in the biocompatible design standard, every principle has a set of objectives. And those objectives are objectives that you have to fulfill in order to make that principle happen. And then, what do you think we do with objectives? We break them down into specific guidelines. Okay, so you have principles that are broken down into objectives, and then the objectives are broken down into guidelines, and the guidelines are specific actionable things that you as a designer can do to make certain that objective is met, right? So we're not going to look at all of the principle 6 objectives. We're just going to look at a couple, because this is the way in which I am thinking about working with microbiomes and viromes to create healthy ecosystems and healthy humans, okay? So yeah, nurture healthy microbiomes and viromes in all ecosystems so that they are providing all of their services, but also providing competitive exclusion. All right, objective 6.1. Yep, this is what it looks like. This is the objective, and then these are the guidelines. There's 7 guidelines underneath this objective. And for this group, I'll actually read them to you, and I'll have you ask questions because they are, as I go through it, hopefully it'll start to build in your mind this idea of how to work with microbes. The thing we do with all of these is that one of my rules for writing all of this is that all of the principles, objectives, and guidelines have to be written in primarily positive language. In other words, I'm avoiding a list of don't do this and don't do that and don't, because it's like, as a designer, so if there's a negative statement, it comes after a positive statement, and it's there to help scope or define that positive statement a little better, okay? So what this is going to be is a bunch of statements of like, this is how to think about it. This is the positive way to engage with it instead of just telling you all the things not to do, okay? So let's see here. Oakland, can you read my main objective for me?
Recognize microbiomes and viromes as essential living ecologies and design technologies, and interventions to preserve their integrity while managing pathological or harmful organisms with ecological precision.
Yeah, so yes, recognize microbiomes and viromes as essential living ecologies. And then, after you've done that recognizing, design technologies and interventions to preserve the integrity of those microbiomes and viromes while managing... Don't poison them while managing pathological or harmful organisms with ecological precision. Don't carpet bomb a microbiome trying to get one bad guy out of there, because all will happen is you're going to open up territory, and the bad guys are going to come back in and recolonize that. Yes.
I don't mean to interrupt you, but I'm just sitting here thinking about the gut microbiome, and is it true that when you have die-off, it's usually the bad bacteria that lives, so it's the good bacteria that's easier to kill off?
The better way of saying it would be to say that for which the environment is favorable will survive. If you have gut dysbiosis, for example, and you do something to kill off a lot, like take an antibiotic, and you get a die-off, what's going to thrive after that are the things for which the conditions are still there. So if the conditions that cause the gut dysbiosis are still there, then yes, the pathogens are going to be favored. So it's not oh, well, I have bad things in my gut, I'll just carpet bomb them with antibiotics and then it'll just fix it. No, what'll happen is you'll knock them down, but if you don't solve the underlying, if you don't change the ecological niche, then the thing that thrived in the ecological niche before is going to thrive in the ecological niche again. Yes, and stronger versions, because what's happened is it's adapted to that particular antibiotic, right? Which is why we're taking the approach we're taking. Because if we take the kill it all, then what we do is we simply breed stronger pathogens, is what we end up doing. Okay, so how about this? Create technologies that expect and partner with the presence of microbial and viral ecologies, actively creating and supporting the environmental conditions that allow them to maintain their ecological function. That makes sense? Okay. Employ strategies such as properly oxygenated conditions, careful selection of materials and surface finishes, the addition of plants and lichens, the presence of broad spectrum sunlight to create an environment that encourages the self-regulation of microbial and viral ecologies. In other words, create a good environment where the good guys can thrive, right? 3, deploy antibiotic, antifungal, and antiviral agents only in extremely narrow and selective cases where human or ecological system health is significantly threatened, preferring instead actively work with beneficial organisms and viruses to restore and maintain safe and ecological health conditions. Avoid, now here's the negative part to scope that, right? Avoid the routine incorporation of antimicrobial agents or biocides into materials, surfaces, or products where their presence can provide marginal benefit while contributing to ecological disruption or acquired resistance. What is that acquired resistance? So you're just talking about that, right? It's like when we take and we use prophylactic antibiotics in cattle, what are we doing?
Breeding better bacteria.
We're breeding better bacteria that know how to get around the antibiotic, right? So this one is saying save those strong antimicrobial and biocidal things for when they like if you have a severe kidney infection that's going to kill you, then heck yeah, take those antibiotics in there and save that person's life. Absolutely, right? Don't use them prophylactically. Don't use them on surfaces, that sort of stuff. Use competitive exclusion where you can and save those powerful agents for when they're really needed, right? Yes.
So another example, like to talk about what you were talking about, like if you have a skin infection,
Yes.
an oral antibiotic is not this strategy. Like that's the carpet bomb strategy.
Right.
So find a topical
Yes.
and keep your microbiome in your stomach intact
Yes.
and help support that.
Yeah.
That guy's not going to waste resources defending against something that isn't on the field of play. So if the
Right.
field of play is natural, you get sort of the natural response, and then you can have those tools available to use.
Yes. Okay, we'll go through the rest of these really quick. The next one is avoid prophylactic use of biocides. We can kind of get that. Where microbiomes or viromes have shifted towards a pathogenic or harmful state, consider using biological inoculation with a consortium of beneficial organisms to help reduce and manage the populations of the specific organisms of concern, attempting to shift the ecology back towards a healthy state. You notice what I'm having to do here. This is written for scientists and engineers and practitioners. So it's written in scientifically precise language, right? So they can accept it. So what we're basically saying here is where the environmental conditions have gotten bad and are supporting the bad guys, then one of your primary strategies is to correct the environment and get a better environment in place so that the good guys can win, right? So makes sense, okay? Then when controlling pathogens, favor interventions that promote long-term ecological and evolutionary stability rather than relying on strategies that might drive rapid adaptive responses, unstable host pathogen dynamics, or repeated cycles of suppression and escape. What did I just say there? I said something very controversial. What did I just say? This is scientific code for be very, very careful with the dynamics of your vaccines. Okay? If you have a vaccine that does not completely eliminate the virus, for example, this is you drive instability in the genetics of those viruses and you breed new viral strains very quickly. Okay, this is why we don't really have a good vaccine against the common cold. It's too unstable. It changes too quickly. If you try to deploy a common cold vaccine, you just drive it farther faster. It's also the reason they have to change the flu vaccine every single year. And then they guess as to which strain is going to be, and if they usually guess wrong or partially wrong, right? And it's the same reason that, okay, I'm going to say it. Sorry, Nicole. It's the same reason that those specific vaccines from, recently, okay, had, they had very complex dynamics, and they actually drove the instability of the pathogen that they were trying to control. Okay, so it's like this guideline tells you to be very careful of that particular dynamic in scientifically robust language that says this can be problematic. Be mindful of it. And then the last one. Reserve sterile or near sterile conditions for situations in which they are functionally necessary, rather than treating microbial eradication as a default strategy for health, cleanliness, or safety. Okay? So we've been trying to convince, this is great, we've been trying to convince a hospital to take this approach. Here's the reason. What do you think the microbiome of a hospital building looks like?
Horrible. Poison.
Best place to get sick.
Right. Okay. So your problem is that if you do sterilization of a surface in a hospital, what's happening is there's reserves of bad guys in all the nooks and crannies, on the fabric surfaces, underneath the chair, in the ventilation ducts, everywhere there's these bad guys, right? And so what you're doing is you're coming along and you're going, I'll fix it. I'm going to sterilize this surface. And so let's say for the sake of argument, you have magic sterilizing fluid that actually does sterilize the surface. That doesn't exist, but just assume it does, okay? Right? As soon as you do it, okay, and you go like this, the last wipe, what's happening? It's being recolonized by all of the rest of the microbiome in the building. Yes?
Do that on repeat, you're actually priming those organisms to be able to recolonize faster and better.
Correct. Right? Therefore, sterilization as a hygiene strategy needs to be used very carefully. Where would I definitely sterilize in a hospital? Well, yes, we're going to put the surgical instruments through the autoclave, because that's going to be reaching down into the exposed internal, area inside the body. So yes, we're going to sterilize that, okay? And if we have a place that's definitely a pathogenic hotspot, right, we might sterilize that. Somebody who's very sick throws up on a surface, we might sterilize that surface, but...
Can we come in after that and with a washcloth from the kimchi and the sauerkraut and the
What we want to do then is immediately we want to get the good guys on the job. What we really want to do in a hospital is we want to establish a very powerful, positive microbiome as much as possible and support that, so we have competitive exclusion. So that we're not having to sterilize, because they'll go through those. So we're going to sterilize the floors, we're going to sterilize the walls, we're going to sterilize every surface and everything. And you're like, yeah, it's just getting worse and worse and worse in here. Yeah, of course it is, right? So I'm trying to get there's a couple of major universities we're trying to get to maybe work with us to actually do the peer-reviewed science, and because I can explain this to you and it makes perfect sense, but nobody in a hospital is going to try this until they have published peer-reviewed papers that they can cite, because the liability is too high.
Yeah.
Right? So we're going to have to, so what it needs to do is it needs to go through peer review, it needs to be published, right? It needs to be recreated, like somebody else needs to replicate it. Then we need to do clinical trials on it and show that it works, and then you'll be able to finally get a hospital to do something.
Anybody
I haven't found it yet. If anybody finds it, let me know.
If it's anywhere, it's probably Russia.
Yeah.
So the issue here is the empty niche, right?
Yes. We're creating an environmental niche that the bad guys can move into. All right. I'm going to go fast now because I've only got a few minutes and we got a lot. See? Okay. I told you I can't clear my throat in less than 90 minutes. Yep. Okay, I'm gonna go do two more really quick. Indoor microbiomes, right? The objective is design buildings and building materials to create and maintain a healthy microbiome environment in the indoor environment to support human health. Okay, how do we do that? Here are the guidelines. 1, connect building microbiomes and environments to the natural microbial and viral ecologies that surround the building, especially by allowing adequate exchange of fresh outdoor air. Yes. There you go. Okay. If we maintain a healthy environmental microbiome outside, then we couple the outside to the inside, right? Establish populations of benign or beneficial microbes in the air and on surfaces in human environments to promote competitive exclusion of pathogenic or harmful organisms. Design buildings to maintain healthy moisture and humidity dynamics that prevent chronic dampness, trapped condensation, or desiccated conditions that destabilize indoor microbial ecologies. In other words, you want it in the Goldilocks spot, not too wet, not too dry. Okay? Yep. Allow variations in humidity within context-appropriate ranges. All right. 4, leverage broad-spectrum daylighting for illumination to also assist in the regulation of the indoor microbiome, providing adequate access to the full spectrum of daylight to discourage the growth of harmful microbial organisms. So there's another one of the principles have to do with this for us to get broad spectrum sunlight, and when we design indoor spaces, how we have to design them so that the windows don't filter out all the beneficial parts of the spectrum that we need, which, by the way, increasingly new buildings do that. If you ever wonder why you can sit next to a building, a window inside of some of these new buildings and still feel like you're getting SAD, seasonal affective disorder, the reason is because it blocks so much of the spectrum, right?
Now it's just efficient coatings.
Right. But we can do better than that. All right.
So what would be the threshold for that?
Huh?
Whenever you talk about that, the percentage of the natural light coming through, what's that threshold where you
So
I
I really like to specify low iron glass with a better than 92% transmissivity. Full spectrum, no low E coatings, that kind of stuff. And we actually have to build the building geometry and shading devices to actually do the thermal environmental control. But it's super critical for human health that we have that full access to full spectrum sunlight inside the buildings.
What wavelength does that go down to for that?
I have a whole tables for that if you want to see them. Like it goes through all of this. It's also in the same standard, all the different wavelengths and how they affect different body systems. I think I sent that. Oakland can probably tell you because I sent that to her yesterday with all the tables for funsies. Okay, and then employ cleaning and maintenance regimes that work with the indoor microbiome to allow it to stabilize, self-regulate, and increase in ecological complexity. Avoid practices that involve chronic use of biocides or routine sterilization. So that's why I make all of my own cleaners, so that I'm avoiding these cleaners that try to sterilize and kill everything. Instead, I try to use cleaners that mechanically clean, that is, get rid of dirt and grime, while at the same time creating the pH conditions and so forth that I want in order for healthy microbes to be able to dominate in that environment, right? All right, so air turnover in buildings. I've got 5 minutes. Here we go. Okay, air turnover in buildings, very important, right? Air exchange continually brings the outdoor microbes into occupied spaces. And then low turnover indoor environments tend to become more biologically simplified and human dominated. Establishing a healthy building microbiome. Okay. Guess what I use? I actually now, when I'm creating a new building, I want to inoculate the building during the commissioning of the building. guess what I use as part of the inoculation for the building? Milk, kefir, and sauerkraut. Okay. Now,
I did it yesterday.
yes. So we have the milk kefir. By the way, I tend to use milk kefir instead of yogurt for 2 reasons. And if you haven't, we have some cups here. So if you want to try a little of the milk kefir,
Oh no,
is that?
that's the grain.
Oh, that's the grain. Okay. They brought me cups, so I was sitting there going, well.
People want to go home with grain and start their own.
Oh, okay.
Oh, you wanted them to try.
No. If you want grain, okay, great. I didn't know you were going to offer that. Okay.
Yeah.
So if you want some kefir grains, then we have some cups and we can get some kefir grains for you, and you can take the kefir grains home and start your own milk kefir. Okay? It turns out that sauerkraut, lactoferments like sauerkraut and lactofermented kimchis and so forth, and milk kefirs have some of the strongest consortia of bacteria, archaea, and fungi that help to establish protective microbiomes in buildings and in our own guts. Part of that is because in lactoferments, we're fermenting in a low oxygen environment, and most all of the human pathogens are anaerobes. And so we can get beneficial anaerobes in an environment where an anaerobic condition emerges, they will tend to help dominate and help control the pathogens. Yes?
I would assume water kefir is going to be in a similar category.
It's similar, but it's not nearly as biodiverse.
Yeah.
The reason is the food source is not nearly as biodiverse. And so milk kefir specifically has this much broader range of biodiversity in its microbiome. Okay. Yes.
How do you apply it? You put it in a spray bottle, or?
I literally just ferment my sauerkraut in the building. What's happening is it's constantly off-gassing, right? And so just bring in gallon jars of sauerkraut that are in the middle of fermenting and put them in different places around the building. Let them off-gas. Okay, same thing. Come in. Usually, if you're doing milk kefir, we'll put a towel or a coffee filter over the top, and we'll let it metabolize. And as you are straining out the milk and the kefir and recovering the grains and putting new milk in every day, because you'll do it every day, you are actually volatilizing, all of this. Now, are we going to be able to develop better ways in the future? Yes, but this is like what you can do immediately, super simple, and you can eat the results, which means that you can actually inoculate yourself and the building at the same time. Okay? So establish that beneficial. Problem with drywall. Okay. So now you can see why I need an hour and a half. It's like a couple hours for this stuff, right? It's like you keep on going. All right. So one of the architects we were working with, she took and did a bunch of samples of the paper backing on drywall. And pretty much every sample that she took had mold spores latent in the paper backing. And so this is why drywall is chia pet for mold. Just add water, right? The reason is that the cellulose in the paper backing is food for the mold. The spores are there, and the gypsum acts as a nice water reservoir. So as soon as it gets wet, it holds the moisture and keeps the moisture on the paper, and the paper is the food for the mold, and ta-da, mold. Okay? This is why I do not put drywall in buildings that I'm specifying. It's like it's just basically mold waiting to happen. Okay, and then clean surfaces without biocides. We kind of talked about that. Like using biologically correct cleaners that maintain the good microbiology. And then the last thing in my last minute is human microbiomes. Design all products, processes to support and maintain a healthy human holobiont. And then you can, if you really want that, you can take a picture of that one and read it, right? But I will just say this. Here is the design relevant human microbiomes, right? Gut microbiome, oral microbiome, respiratory microbiomes, skin microbiomes, urogenital, ocular, and then there's the maternal early life microbial transfer pathways, like vaginal delivery and so forth. Let me hit this really fast, Jake, and I'll come back to you. Okay. And then representative documented taxa across major human microbiome domains. This shows some of the major documented taxa of bacteria, archaea, fungi, viruses, and protozoans and other microeukaryotes that show up in some of those microbiomes. You can see how biodiverse they are. And those are just the ones that are documented. They're actually much more biodiverse than that, okay? So this is where we, okay, this is a diagram of what happens when you have good gut bacteria and good gut microbiome, okay? This is what happens when it's bad. All this research is very extensive. And I could tell you stories about people who've come to me with gut dysbiosis that were, I mean, yeah, the whole problem is the medical industry didn't know how to do with them. Made it worse. They made it worse. And then by actually treating the root cause of gut dysbiosis, we were able to get them to spontaneous. I didn't heal anything. They healed themselves just by letting their gut get into the conditions where their microbiome could recover.
Can I make you go too long by telling a story about that?
Yes.
So I got Giardia, and I went to the not knowing I had it. Went to the doctor, and he figured it was Giardia based on the symptoms. And he said, but it could be E. coli. And he said, I don't know which one it is, so I want to test because if I give you antibiotics, it would kill everything. You're going to die. And he sent me home with a protocol of prebiotics, probiotics, and all of this other stuff. So he attacked it from the gut microbiome approach, which is something I've never experienced.
Yes.
He also retired last December. But yeah, I know. But I was like, what?
I have a lot of stories.
That's what I was gonna do. But yeah.
On that, you start to see that the gut microbiome has a lot to do with obesity, diabetes. There's a huge amount going on there that, again, we could break. When we get into this with medical practitioners, we break all this down in detail. Here is an example of beneficial versus bad lung microbiomes and what happens when you have good and bad lung microbiomes. Okay, because this is, again, I want you to thinking about more than just your gut. It's like you got your lung and your skin and your nasoresonal and oral cavities and all that kind of stuff, right? And then birth. I need to say this before because this is super important. Okay, so birth mode influences early microbial establishment, right? This is critically important, and this is something that is now, the literature is now coming out on it, but we've known this for a number of years. Vaginal birth exposes infants to maternal microbial communities during delivery. What happens is, as the baby is delivered through the vaginal canal, the mother's microbiome is actually exposed into their oronasal cavity, and this starts to actually establish their skin microbiome, their oronasal microbiomes, and their gut microbiome. There are certain parts of the biodiversity of the gut microbiome that if you do not get them from your mom at the moment of birth, they're hard to get, like the bifido strains. Okay? And so it's critically important, and this means that the health of the mother's microbiome has a massive impact on the health of the child. So one of the things we need to be doing is looking at how to support the mother's microbiome health, especially vaginal microbiome health, as she is going through pregnancy towards delivery. Very important, okay? Now, early colonization helps shape immune development and later microbial trajectories. This has prompted research into microbiome restoration strategies after C-section, although best practices are still under study. In other words, if you deliver a child via C-section, it misses that pass through the vaginal canal and that initial inoculation. So the question is, it's now not controversial that is a critical miss, and it fails to jumpstart the baby's immune system properly. Okay, because 70% of the immune system is going to be like microbial. So how do you restore that? And so it's like there's controversial, like what is the exact right way. There's been things as simple as vaginal swabbing that then is then like, but then of course there's people like, oh, what if the mother has, certain forms of dysbiosis and we don't want to expose that to the child. And so it's like, that's why I have to technically say that it's still under study, right? We just know it needs to be done in a good way. We know that initial seeding of the infant's microbiome. And so what I tell everybody these days is if you have an OBGYN that denies this reality, I would be careful of that OBGYN or ask them to go read all the latest research, right? It's like if we have to do an emergency C-section, then how are we going to make certain that the infant actually gets, I know this is going to open up an hour's worth of questions, but yes, real quick.
There's also the case when you have a vaginal birth, your cervix completely remodels.
Yes.
So if you have a inhospitable vaginal, there's also the uterine microbiome. It actually resets, like God or nature gives you a second chance. It
Yes.
clears all HPV. That's why we don't test now for HPV until post childbirth age, because if you give birth vaginally, it erases it.
Yeah, there's all these dimensions.
Creates a brand new organ. So we've done biopsies on cervixes pre-labor and post-labor, and it's a completely new organ.
Yeah. Yeah. There's all this stuff that's like, okay, now I got like 47 questions. I tell you what, Jake, hold on just one more second.
I'll take a break and they could mob you, and then we can start on time again.
We're almost there. Let's go.
Were you on Alex Zach's? I think you were.
What was that?
Were you on Alex Zach's podcast, The Way Forward?
No.
Okay.
No.
Someone was on there, and he's recognized this and talked about it, and he's starting a consortium of practitioners who recognize this and are working with women.
Yes.
And it's called The Way Forward.
Okay.
Yeah.
I'll have to look that one up.
I'll see if I can find one.
To inoculate a C-section baby, all you have to do is stick your finger in your crotch and then put it in the baby's mouth. And it's pretty easy. And then avoid that afterbirth wash of the baby. Don't let them wash your baby clean of everything that they have on
I
them.
agree. The whole problem is I got to teach this to medical practitioners. Right. Right.
You know.
Again, the challenge is liability in hospitals. And so we got to get something that they can feel that they can do without accruing liability.
And it's also, it differs by status, because our vaginal microbiomes differ extensively by status. So the NIH has known this for over 10, 15 years.
Yes.
We can't figure out how to put it into implementation because of the disparities.
Exactly. And it comes down to that. It's like, how do you, like, hopefully in the vast majority of cases, if we had healthy women, then their microbiomes are fine, and you could do exactly that. But there's like, what about the 10% of cases where that's not the case, and now you've introduced something to the infant that's problematic? You got liability, but also you have health issues that could be. So it's like, that's what I'm saying. Best practices are still under study. That's probably the most I can say at the moment. Let me finish up. Colostrum, make certain that the infant gets the colostrum within the first hour. Boy, I could say a lot about that. That is also an immune system primer, but it also starts to prime the microbiome. Okay, very important. And breastfeeding transfers and feeds the beneficial microbiome. It transfers maternal microbes to the infant during repeated daily feeding, helps shape early microbial colonization of the infant's oral and gut microbiomes. Breast milk also contains human milk oligosaccharides that the infant cannot digest directly, but that selectively feed beneficial microbes, especially the bifidos, helping stabilize the developing microbiome. Okay, so, and ongoing, the mother's breast milk also, we now have increasing evidence is actually the mother's immune system is much more mature and is able to create antibodies, and the milk will transfer antibodies to prime the infant's immune system with antibodies that the mother's immune system has been able to create, that the infant's immune system is not quite mature enough to create yet. This is why vaginal delivery followed by colostrum followed by breastfeeding, that trifecta, if you can get it, is nature's way of getting the infant's immune system like optimally charged, provided if the mother's immune system is healthy, right? So that fair enough to say? Cool. Jake.
So how do I know if my house is environmentally is there a test or analysis?
How fast does the food in your refrigerator spoil? At my house right now, we tried an experiment and we could not get the food to spoil. It stayed 2 weeks in the refrigerator and it would never go off.
I mean, you got a dog and a cat. You got a 100-year-old schoolhouse. You got HVAC. You're using across-the-counter pharmaceuticals.
Yep. How long does it take the bread to mold? How long does the food to get? That's like short of testing. That's one really good way. We're going to take a break now. I know we are over. Let's take a break, and then we'll come back, and I'm going to reset and hit EMFs and RFs.
Yeah, we need to refill the water.
Is this slide deck available anywhere? Can you send it out?
I guess I could. It's like I just created